So our next subtopic is procedure of agarose gel electrophoresis. We'll discuss how we are able to run gel electrophoresis. First of all, how gel is casted, how gel is prepared. First of all, gel is prepared by dissolving agarose powder as per requirement in some suitable buffer. If we want to separate smaller molecules, we use higher concentration. If we want to separate larger molecule, then we use low concentration of gels. Then it is heated, that agarose is melted in the buffer. The melted agarose is allowed to cool to some extent before it is poured into a cast. And then after pouring, a comb is placed in the cast so that wells are made where samples are loaded with the help of micropipettes. Finally, place the gel at 4 degree or at room temperature till it is completely solidified before electrophoresis. Then how samples are loaded? Here, once the gel is solidified, the comb is removed for loading DNA samples. When we remove the comb, the wells are formed in the gel, and in these wells, samples are loaded with the help of micropipettes. Loading buffer is mixed with samples to increase density so that the samples are submerged at the bottom, and similarly some dye is also added, like bromophenol blue, which gives some color so that the bands or the moving DNA molecules can be seen. The gel is placed in the electrophoresis unit and is covered with the buffer. And similarly, samples are loaded in the valve, as we have discussed earlier on, with the help of micropipettes, and electrophoresis is carried out at the suitable voltage. It can be slightly faster, it can be slightly slower, depending on the need of the procedure. Electrophoresis हम कैसे कैरी आउट करते हैं? हम हॉरिजॉन्टली भी कर सकते हैं, वर्टिकली भी कर सकते हैं. अगर आप जेल को हॉरिजॉन्टली अलाइन करें तो इट इज़ हॉरिजॉन्टल, अगर आप वर्टिकली अलाइन करें तो इट इज़ वर्टिकल. नॉर्मली हॉरिजॉन्टल जो इलेक्ट्रोफोरेसिस है इट इज़ प्रेफर्ड, इसलिए कि जो सेपरेशन होती है वो यूनिफॉर्म होती है. बफर यूज्ड इन द जेल्स इज सेम एज द रनिंग बफर इन द इलेक्ट्रोफोरेसिस टैंक. द जेल इज रन normally at 80 to 150 voltage until the dye is about 70 to 80% down the gel. Afterwards, the gel is removed from the gel box and it is placed under some UV source where DNA fragments are visible due to presence of ethidium bromide. Size of DNA fragments they can be estimated with the help of DNA ladder. I have earlier discussed that in one of the well, rather in the first well, normally DNA ladder is loaded and we compare our separated strands of DNA with the bands of ladder, and in this way we are able to estimate the size of the DNA bands.